Good afternoon and welcome back. This is Dr. Sanjeev Pandey and you are watching SP at Bio, an educational channel. In this video lecture, we will talk about bacterial flagella. As you know, flagella are locomotory organs and because of which an organism can move from one place to other. It can sense the food and it can move towards food, or it can repel itself and can move away from it. So many bacteria possess flagella. How does the bacterial flagellum looks like? How it is different from an eukaryotic flagellum? We will talk about that. So first of all, the bacterial flagellum is a locomotory organ of the bacterial cell, and it accounts for 2% of the dry weight of the cell. It is, of course, an elongated structure, and one to many flagella may be present on a cell. And depending on that, this feature is called, this position and number of flagella, when you talk about them, this is called flagellation. So the flagellation in bacterial system are of different types. It may be afflagellated altogether, that is called amphitrichous. The condition is called amphitrichous. It has not been shown in the slide here. Only those has been shown which poses flagella. So the first one is the monotrichous. Mono means single, trichome means the flagella. So monotrichous, they have a single polar flagellum, of course, on one pole. It may be at a different region of the cell, according to which the name might change. If it is present in this area, it is cephalotrichous rather. But in most of the cases, when it is cephalotrichous, it is more in number. So Pseudomonas aeruginosa and Vibrio cholerae, they show monotrichous flagellum of this type. Lophotrichous, when a tuft of flagella, a group of flagella are present on a single pole, like in Pseudomonas fluorescens. And it is called amphitrichous, as the name suggests. Amphi means both, when the flagellum is present on both the ends, both the polar ends. It may be single or it may be in tufts. In both the cases, it will be called amphitrichous or amphitrichous. The example is Aquaspirillum serpens. Spirillum serpens is also amphitrichous, like this. And if the flagellum is present all around, it will be called peritrichous. Peri means the periphery, or it means surrounding the cell. So if the flagellum is flagella, many flagella are present surrounding the cell, like in Escherichia coli, Salmonella typhimurium, like this, then the condition is called peritrichous. Now let us see the structure, the ultrastructure of the flagellum. Here is the gram-negative flagellum, and this one is the gram-positive flagellum. We will talk more about gram-negative because it has been described the most. However, gram-positive flagellum are also described, but they are very simple compared to the gram-negative one. So the gram-negative flagellum, this one is the flagellum, and this is the cell envelope, gram-negative cell envelope, which comprises of three layers. This is plasma membrane, this is periplasmic space, this is the peptidoglycan layer, and this is the outer membrane layer. It also contains the lipopolysaccharide layer. So the gram-negative bacterium have a flagella that consists of three parts. The filament, the upper part is called filament, this part is called the proximal hook, and the part which is embedded in the envelope is called basal body. The filament is the most thinner and most elongated part of the flagella, and that is responsible for moving the flagella, taking the buoyancy of the flagella from the environment. But the whole structure is important. If rotation does not take place by the basal region, then flagella will not rotate. That means filament is the thinnermost part. It comprises of a protein called flagellins, which are 20 to 60 kilodalton proteins, and this one is most elongated part of the flagellum, 6 to 8 micrometer. It may be 10 times longer than the cell. There is a terminal cap in these flagellin proteins are arranged spirally in most of the cases. Sometimes they may be cyclical also, and the terminal position is occupied by a protein that is called HAP3 protein, hook associated protein 3. and that has been named as hook associated protein, initially thought to be hook associated, but later on it was found to be on the cap protein. So proximal hook is this region, which is comparatively a short region of 0.5 to 6.6 micrometer long, but it is wider than the filament and also the rod element, the basal element. So it comprises of two types of proteins rather. There are other proteins also, and principally these two types of proteins, HAP1 and HAP2, hook associated protein 1 and hook associated protein 2, are present in this hook region. And hook region is little bit bent in gram negative, also in gram positive bacterium. Now talking about this basal body, which is most complex structure, it comprises of a central rod element, and there are four rings on this basal body. These rings are named according to their positions. Like this outer ring is called the P ring because it is L ring, not P ring. Here the P should come after L. This is L ring because it is positioned at lipopolysaccharide layer, and then the second one is the peptidoglycan P ring, peptidoglycan ring. The third one you can call them together because they are mostly associated, the SM ring or the M ring and the S ring. M ring is the membrane ring, and the S ring is supramembrane ring. Both are juxtaposed one above the other. The S ring is comparatively smaller than the M ring, and there is a central rod that acts like a shaft of a motor inside these rings. And this part of the ring, basal body, is motor. It is responsible for the rotation of the flagella. We will talk about them little later. And there are many proteins associated with this SM ring. The particular protein which surrounds this SM ring is called Flag, and that is surrounded by, flanked by two different proteins which are motor proteins, MOT A. This one is MOT A and this one is MOT B protein. MOT A and MOT B protein, and there are two other proteins, Fli M and Fli N. Those are hanging towards the cytoplasm, so they are called C ring also by another nomenclature. So there are altogether 5 rings: L ring, P ring, S ring, M ring, and C ring. This region of the flagellum is very important because it is responsible for flagellal rotation. We will see about them. So how does it differ from the gram-positive flagellum? Gram-positive flagellum is comparatively simpler because there is no outer membrane, and there are 2 rings only, the M and the S ring. The hook region is bigger, it is longer, and it is more wider than the gram-negative counterpart. And there are no P ring and L ring sort of things because the peptidoglycan layer is thick. It holds the hook region. The hook region arises from the S ring, and it circumpasses the peptidoglycan layer and little bit exposed, and after that the filament is present. The hook is little bit like a funnel in case of gram positive flagellum. Sorry. Let's move to the next. So this is a sort of flagellum. This is another kind of flagellum called endoflagella or periplasmic flagella, and it is a special characteristic of a bacterium spirochaete, and it helps in spiral rotation. It is hidden inside the periplasmic space, you can see, and it is spirally oriented like this on the cell, and there are axial filaments which actually slide upon each other, because of which the bacterium shows corkscrew like movement on the surface on viscous medium. So this is another kind of flagellum. It is not an exterior flagella, rather it is hidden. It is called endoflagella. And now we will talk about the flagellal rotation, how it occurs. The flagellal rotation is mainly based on the motor part of the flagella. There is this the S and M rings and the shaft, that is the central rod, and the proteins, Flag, Flag-M, Flag-N, and MOT proteins are responsible. Mainly the Flag and the MOT A and MOT B proteins are responsible for this rotation. It is a proton driven pump, And these MOT A and MOT B proteins, they act like half channels. MOT A and MOT B proteins act like a half channels, means they are so oriented that when protons pass from MOT B through MOT A, if this the flea G has to be oriented in such a way that the proton passes through a channel, a pore-like structure channel, and to align that channel, it has to rotate and position itself with respect to mod A, mod B, and also with respect to mod A. So these two mod and mod act like a half channel, and in between is this flea G protein, and each of these three has to be aligned in a way, and for alignment when one proton passes, because of the motion this will rotate further and it will disalign. So to align again, the next flagella has to come to take the same position, and while doing so the flagella will continuously rotate in one direction in a cyclic manner, and therefore the central rod, that is shaft, will also rotate, rotating the filament and each of the hook and filament, every part of the flagellum, and the bacteria will move or will stop or will tumble and will switch back to its other position. So this is the way how the flagellar rotation in bacterial system occurs. Now let us talk about the difference between the prokaryotic and eukaryotic flagella. This is the last slide, and this is a picture which shows an eukaryotic flagellum, and you can also say cilia. They are equivalent to cilia. So eukaryotic flagellum have two parts mainly. One is inside the membrane within the cell, that is called the basal body, you can say, or they are the flagellar root, and the exterior part, that is the filament-like exterior part. So both have them have a different anatomy, you can say. If you take cross section in the flagellar root region or basal body region, you get microtubules. In the arrangement of these three microtubules are arranged in sets of three. There are three sets of microtubules, and there are nine in a particular ring. And there is no central rod, central ring in this in the basal region. The central rod, that is the two other rings that come in the middle when the flagella is it arises from this cell, it emerges from this cell. So this is a 9 plus 2 arrangement, and this is 9 plus 0 arrangement. But here the 9 surrounding microtubules are in pairs. There are dynein proteins, and these are some spokes-like structures. So we will talk about the eukaryotic flagella separately in a separate video lecture, because it needs a detailed study. And here only I want to show you the difference between these two prokaryotic and eukaryotic flagella. For that, we need to have a glance at the anatomy of this. And this flagellum is, in fact, to some extent, it is surrounded by the membrane, and after that it is exposed. That is, the exposed region is called the end piece, and the membrane surrounded region is called exonym. So these kind of things are absent in bacterial system. So let us talk about bacterial flagella. They are made of flagellin proteins. The principal protein we are talking about. There are other proteins also, but the principal protein is flagellin. Here is also the principal protein is tubulin, and the arrangement is 9 plus 2 or 9 plus 0 arrangement. Here the arrangement is spiral, helical, spiral, or you can say it is cyclic also in some cases. And there are, it can be divided in bacterial flagella, can be divided into three parts: filaments, hook, and basal body. Here you can divide it into two parts: the outer region, outer portion, and the flagellar root. If you divide in a different way, then you can divide it into three parts again: the end piece, the exonym region, and the basal body or the flagellar root region. So this way you can distinguish between the portions of the different portions of the flagella. The movement of the bacterial flagella is proton driven, that we saw a little earlier. Here this movement is ATP driven, and the way the flagella moves or rotates is different. It is rotational in case of bacterial flagella, whereas it is in the form of a wave or beating movement. There are different kinds of beating movements in case of cilia or eukaryotic flagella. So that's all about this lecture, video lecture, bacterial flagella. I think you were benefited with this lecture, and if so, please subscribe the channel and press the bell icon to get further notification. And also don't forget to press the like button, give a thumbs up to the lectures to support future video lectures to come. Thank you very much.